I want to thank you for inviting me to participate in your meeting. I had visited Apollo when I was in India about 6 months ago, giving a lecture in Delhi and then going to Hyderabad. I was very impressed with the Apollo program. We're trying to develop a collaboration to create a group of diagnostic tests and biomarkers to predict cardiovascular diseases, and we're sort of in the midst of that planning. I have, as you've heard, been interested in research since 1958. So it's been, 62 years. And I have fortunately worked with some outstanding professors and trainees and advisors. And I began working with cyclic AMP in 1958 and discovered that many hormones and molecules regulated cyclic AMP accumulation in tissues to produce a variety of biological effects. After about 10 years of working with cyclic AMP, I became interested in cyclic GMP, guanosine monophosphate. It had been discovered in the late 1960s, and that interested me. And I thought perhaps cyclic GMP could be another intracellular messenger. And as I began working with cyclic GMP, that led to the discovery of nitric oxide in 1976-77. Our first publication with nitric oxide was in 1977. Today, there are about 160,000 publications in nitric oxide. It's just gone in all sorts of directions in biology and medicine. And it's permitted many of us to discover some very interesting drugs to treat a variety of diseases. So what I want to do in this lecture is give you an overview of how cells talk to each other. We call it cell signaling, and they do it with messenger molecules. I will probably eliminate some of the biochemistry. I don't want to lose you with too much detail in the biochemistry. And then I'll concentrate on, towards the end of the talk, on the clinical applications, where this is going and why it's so exciting. And to see one's basic research have such broad application in clinical medicine is very rewarding and very exciting. We know that cells communicate with each other. They talk to each other. They have to. This is a cartoon of how this process works. This is a picture of 3 different cells. I'll call cell type 1 a neuronal cell. Cell type 2, I will call an endothelial cell. And I'll call cell type 3 a vascular smooth muscle cell in the wall of that vessel. And they're going to communicate with each other. And they do it by creating messenger molecules. If it's a neuronal cell, we call these messengers neurotransmitters. If it's an endocrine cell, we call these messengers hormones. If they're macrophages or lymphocytes, we call them growth factors, cytokines, chemokines. These substances are released into the bloodstream. If they're small molecules, they often bind to a transporter or carrier to protect them from degradation and metabolism. If they're large molecules, as large peptides or even proteins sometimes, they don't usually have transporters. But when they're released from the cell, they go searching in the body for their target. And they identify their target cell, in this case an endothelial cell, with a protein on its cell surface. We call those proteins receptors. So the messenger molecule binds to this receptor in a 3-dimensional conformational fit, like a key in a lock. Only the right messenger recognizes the right receptor. That's where the specificity in the communication takes place. The first such messenger discovered inside of the cell, we call them second messengers. When the ligand binds to its receptor, it perturbs the membrane and biochemistry to make intracellular second messengers. And while there are dozens and dozens and dozens of these first messengers, there are only a modest number of intracellular second messengers, only 5 or 6 so far. Now, most of these messengers have had broad application in medicine, and many of them have been recognized with Nobel Prizes. The first intracellular messenger was cyclic AMP, discovered by Earl Sutherland and Ted Rall in 1957, the year before I joined their lab as a student, medical student, graduate student. Today, there are another 4 or 5 messengers: calcium, cyclic GMP, the eicosapentanes, the prostaglandins, nitric oxide. So there are only a few.
Yeah.
Nitric oxide is a very unique intracellular messenger because it's a gas and it's a free radical. And when we discovered a free radical gas being a messenger, that was unique and attracted the attention of the Nobel Committee. The nitric oxide can do work inside of the cell, or it can come out of the cell and regulate the biology and biochemistry of other cells, adjacent cells. In this cartoon, nitric oxide comes out, goes over to the smooth muscle cell in the wall of that blood vessel, and increases the production of cyclic GMP to produce various biochemical and biological effects, including relaxation of that tissue. Okay? Next slide. As we were examining the production of cyclic GMP in heart extracts, liver extracts, brain extracts, we accidentally discovered that a couple of small molecules activated the enzyme. There's several isoforms of guanylate cyclase, but the soluble isoform is the one that's regulated by nitric oxide. And when we found these substances increasing cyclic GMP, I thought that cyclic GMP would antagonize the effects of cyclic AMP, and I knew as a student that cyclic AMP would relax smooth muscle, airway smooth muscle, gastrointestinal smooth muscle, vascular smooth muscle. And I thought cyclic GMP would cause the opposite. So at the time, we were working with tracheal smooth muscle from beef, a very nice, clean system, very homogeneous with smooth muscle cells. When you do biochemistry, you can't do biochemistry in mixtures of cell types because you don't know which cells are making your products and which cells are responding physiologically. So we developed this tracheal smooth muscle model. We put azide, sodium nitrite, hydroxylamine on smooth muscle cells, and they increase cyclic GMP as they did in other tissue. I thought the tissue would contract, but surprisingly, it relaxed. That was a big surprise. So now we have 2 messengers, cyclic AMP and cyclic GMP, that's related to smooth muscle relaxation. And then I remembered, as a house officer, giving nitroglycerin to patients with heart attacks in the intensive care unit to lower their blood pressure, to decrease afterload and protect the heart and let it heal quicker. So we put nitroglycerin on these smooth muscles. We knew they were relaxant agents with vascular muscle and other smooth muscles, and it increased cyclic GMP. And then we discovered that there were many other nitrogen-containing compounds that would do the same thing. They would increase cyclic GMP. They were relaxing smooth muscle, and we coined the term nitrovasodilators. And it included nitroglycerin, nitroprusside, inorganic, organic nitrates, and nitrates. And that then really got me very excited because we now had a system to regulate this intracellular messenger and follow biological effects with a variety of important drugs, obviously. And I thought that by understanding this whole biochemical pathway, I could discover some new and interesting and important drugs. Next slide. There are 6 isoforms of the enzyme guanylate cyclase. The soluble isoform is the receptor for nitric oxide. That's how it works. The enzyme has a heme prosthetic group. Nitric oxide binds to the ferrous iron in the heme. It pulls the heme off of the enzyme out of the pocket, permitting activation of the enzyme several hundredfold, and the enzyme converts GTP to cyclic GMP and pyrophosphate. A reaction very, very similar to the adenylate cyclase pathway that converts ATP to cyclic AMP and pyrophosphate, which I'd studied as a student. The cyclic GMP or cyclic AMP inside of cells at steady state concentration is regulated by its rate of formation and also the rate of its degradation and removal. And there are a family of enzymes, cyclic nucleotide phosphodiesterases, that hydrolyze the phosphodiester bond to form the corresponding 5' monophosphate, 5' GMP or 5' AMP. Those enzymes I knew were regulated by methylxanthines, caffeine, theophylline, theobromine, and that's how I predicted that these drugs could be useful in penile erection. I'll tell you more about that story shortly. We thought about drugs like nitrovasodilators and phosphodiesterase inhibitors 15 years before Viagra came along. I should have pursued it and patented it, but I didn't think it was a major medical problem. So we went off in other directions in the cardiovascular area, in neurosciences, and stem cells, and cancer. These messengers, cyclic AMP, if we can go back, these messengers, cyclic AMP, cyclic GMP, activate a family of enzymes called protein kinases. These enzymes transfer the terminal phosphate of ATP to a protein substrate. There are 100 or 200 protein kinases. Some are regulated by calcium, some by cyclic AMP, some by cyclic GMP. And there are many, many proteins that get phosphorylated. And when a protein gets phosphorylated on its serine or threonine residue, it changes its conformation. If it's an enzyme, it can be activated or inhibited, and that then produces a variety of physiological, biological effects. Okay, next slide. Let's skip this slide. Let's skip it. So let's skip this. I don't want to get into all the detailed biochemistry. Next slide. Okay, so I told you that azide, hydroxylamine, sodium nitrite activated guanylate cyclase, which then they were converted to nitric oxide, which then activated guanylate cyclase. And we had compounds that would mimic the effects of azide. Nitroglycerin, nitroprusside, nitrosoureas, and others, they were all different structures. And I reasoned that they had to make this molecule that we didn't know about. And it was a mystery as to what it was. And we finally concluded, because hemoglobin was an inhibitor of this pathway. Hemoglobin has a high affinity for nitric oxide, which I remembered from my college days in chemistry. We predicted that the intermediate was nitric oxide. So December of 1976, we generated nitric oxide in the fume hood, mixing sodium nitrite, sulfuric acid, and ferrous sulfate. We took the gas, ventilated it into our cyclase incubations, and sure enough, it activated all of our soluble guanylate cyclase activation. That was the eureka moment, really an exciting day. And we said, oh my goodness, a free radical in gas can activate an enzyme. Let's figure out how it does it. And then we spent a lot of time working out the biochemistry and discovering a lot of drugs along the way. Next slide. This is how nitroprusside works, but let's skip this. Let me show you acetylcholine. You're interested in endothelial dysfunction. We predicted in 1978 that nitric oxide was the mechanism of action of nitroglycerin and other nitrovasodilators. When you find an endogenous substance doing something of interest, you have to ask yourself, is this normally present in the body? Do hormones and other agents regulate the formation of cyclic GMP and nitric oxide to produce a variety of effects? A prediction, a hypothesis. Turned out we were right. People didn't believe us for about 10 years. That was a little frustrating. Next slide. Next slide. This is, let's go back. Go back one to acetyl. Here, acetylcholine is an endothelial-dependent vasodilator. Okay. Furchgott had worked on this for a long time. He found that when he put acetylcholine on blood vessels, it caused relaxation, but only if the endothelium was intact. If the endothelium was damaged or absent, it wouldn't work. When he gave that lecture, he discovered endothelial-derived relaxin factor coming from the endothelial cells to regulate the vascular smooth muscle. He presented that lecture in Virginia. I was in the audience, and afterwards I said, Dr. Furchgott, come to my office. I think you're working with a reactive species, maybe a free radical, maybe nitric oxide. Let's collaborate. Well, the collaboration didn't occur. His wife developed cancer. He returned to New York. I moved to Stanford. But a year or two later, we decided to repeat the studies that I was proposing to Furchgott, and this is an example of it. Acetylcholine increases cyclic GMP and causes relaxation in blood vessels for aorta segments. It doesn't affect cyclic AMP. Next slide. Next slide. Okay, this cartoon is of a blood vessel. The endothelium on the left, the smooth muscle in the wall of the blood vessel on the right. If you add up all the endothelial cells and all the veins and arteries, it's a huge organ. It's as big as your brain or heart or liver. It's a big, big organ. We used to think it was very inert. It didn't have much biochemistry or biology. It just influenced the transport of molecules from a bloodstream to the blood vessel to the tissues. That's not correct. We now know that the endothelium is a very, very active tissue. It produces lots of factors, lots of messengers, lots of growth factors that influence blood clotting and all sorts of other things. So let's start on the right-hand side with nitrovasodilators, nitroglycerin. It's converted to nitric oxide. Some of them, what nitrovasodilators do this spontaneously. Some of them, such as nitroglycerin, requires an enzyme to take the NO off that nitrate group on the glycerol backbone of nitroglycerin. The nitric oxide activates the soluble isoform of guanylate cyclase in the top right-hand corner, converts GTP to cyclic GMP. That activates a protein kinase, which phosphorylates proteins, and it phosphorylates many proteins. And you can monitor this on gels with P32 incorporation of these proteins. And some of these proteins regulate calcium concentrations inside of the cell, and they decrease calcium. We knew that myosin light chain kinase, which phosphorylates myosin to influence the latching of actin-myosin filaments, is a calcium-dependent enzyme. And by lowering calcium, you decrease the activity of that kinase. The myosin becomes dephosphorylated, the filaments slide apart, you get relaxation. If you phosphorylate myosin, they slide together, latch, you have contraction. So we figured out the biological pathway of messengers related to smooth muscle relaxation, airway smooth muscle, GI smooth muscle, vascular smooth muscle. On the left-hand slide is the endothelium. Furchgott was studying endothelial-dependent vasodilators, acetylcholine, histamine, thrombin, and others, and they interacted with the endothelium to increase calcium, to permit arginine, an essential amino acid because our body doesn't make enough, we have to have some in our diet, and it converts arginine to endothelial-derived relaxant factor, which is a reactive species that turns out to be nitric oxide. It then goes over and does exactly what nitrovasodilators do through the same identical pathway. So they work the same way, but they're using different receptors in different cells to produce this phenomenon. There are other messengers that regulate cyclic GMP production, the atriopeptides, AMP, BMP, CMP. We discovered that they activate some of the particulate forms of guanylate cyclase, not the soluble forms, but only the particulate forms. And when they increase cyclic GMP production, they also cause vascular relaxation. They influence the renal function, the blood pressure gradient across the glomerulus, filtration. It has effects on the tubular urine as well. Next slide. Next slide. Next. There are a couple of other investigators in the field that have done important work, but I don't have time to review it. The enzyme that makes nitric oxide from arginine is called nitric oxide synthase. There are 3 isoforms, which means they're produced by 3 different genes, but they have very similar biochemical properties, and they have very, very similar structures, but they're located in different parts of the cell. For example, NOS3 is the endothelial isoform that we purified, and it's both soluble and particulate. NOS1, we first purified from cerebellum in the brain. It's primarily soluble. NOS2 is an interesting one because its gene is induced by pro-inflammatory cytokines, endotoxin, IL-1, TNF-alpha. They regulate NF-kappa B, which influences the expression of NOS2. So normally tissues don't have much NOS2, but you find a lot of it in cancer cells, and you see a lot of it when the tissue is inflamed or is infected with gram-negative organisms that produce endotoxin. So it's become an important biomarker of inflammation, an important biomarker of cancer, pre-cancer. Next slide. This is the pathway. This is a stick figure on the left of arginine. The enzyme has some very important cofactors. It requires oxygen, it requires an ADPH, it requires flavines, FMN, FAD, tetrahydrobiopterin, and ADPH. By knowing the cofactors that help the activity of the enzyme, that is very important discovery that we made. Because now we can regulate the activity of that enzyme by regulating the cofactors. That, I will tell you, plays a major role in endothelial dysfunction. The enzyme oxidizes the guanidino-nitrogen to a hydroxy group. That is converted to citrulline and nitric oxide. Next slide. That's how the enzyme works. Next slide. Okay, let's go back one. Let me put this together. By studying the pathway and the biochemistry and knowing what each of these enzymes do and how they're regulated, we can now come along with chemical libraries looking for chemicals that either activate that step or inhibit that step. And that's how we discover drugs. Here's the pathway. Ligands bind to the receptor. They increase the conversion of arginine to nitric oxide. That makes more cyclic GMP. That regulates a kinase. It can be removed by phosphodiestrases. So, but in the next slide. Next slide. Nitric oxide prefers, go back one, please. Let's go back one. Nitric oxide prefers to make cyclic GMP, and that's very beneficial in lots of tissues. However, it's pulled off in other directions. It can be oxidized to nitrite and nitrate. Those become biomarkers of the activity of NOS to make NO. You measure nitrite, nitrate in the serum or urine, it tells you you're making a lot of nitric oxide in tissues. It can form complexes with other metals, transition metals, besides iron, zinc, copper. It can nitrosylate thiol groups. It latches on to the sulfhydryl group on proteins, and there are hundreds of proteins that get nitrosylated. Some of them are very important. Some are receptors. Some are regulate transcription. An important reaction is the interaction of the nitric oxide free radical with another free radical, superoxide anion. Whenever you have inflammation, you're going to make a lot of NO and you're going to make a lot of superoxide. And now they combine to form a very toxic molecule, peroxynitrite, that nitrates proteins, and if they're enzymes, it destroys their activity. So nitric oxide can be very beneficial or it can be very toxic. So it depends on where you're making it and how much you make, and is it in the presence of superoxide. So you don't want superoxide around. You don't want reactive oxygen species because you'll inhibit the good effects of nitric oxide through cyclic GMP. Next slide. As you all know, there's a disorder called endothelial dysfunction. Patients with hypertension, diabetes, atherosclerosis, tobacco use, and probably obesity have blood vessels that do not make enough nitric oxide. And we can measure that by looking at nitrite, nitrate in serum and urine, by looking at cyclic GMP in serum or urine. It's a monitor of nitric oxide and cyclic GMP production. We know that in these disorders, you create another metabolite of arginine. It's an asymmetric dimethylarginine, which is a competitive inhibitor of nitric oxide synthase. It competes with arginine for the catalytic site. It's a competitive inhibitor. If it's competitive, it means if you come along with the right substrate, arginine, you can overcome the inhibition. Because of that, that's led now many companies to create arginine supplements to increase nitric oxide formation. We know that these disorders are associated with reactive oxygen species because they're inflammatory diseases. You make a lot of superoxide. You oxidize the cofactors. And if you oxidize the cofactors, the enzyme doesn't make NO. Tetrahydrobiopterin is very, very critical for enzyme activity. If you oxidize the tetrahydrobiopterin to dihydro, the enzyme now makes superoxide instead of making NO, the wrong thing to do. So again, by understanding the detailed biochemistry, the nature of the enzymes, how they're made, how they're expressed, where they're located in cells, and what their cofactors are, we can now treat a lot of important diseases, including endothelial dysfunction. Next slide. I have 2 more slides to give you a spectrum of where this field is going in terms of clinical medicine and biology. We know that nitric oxide is a neurotransmitter. There are nerves in the brain, the cerebellum, in your gut, in your kidney, and in your penis, in the corpus cavernosum, that release nitric oxide as their neurotransmitter. It plays a role in the vitreous pressure of the eye and glaucoma. It has effects on the retina in terms of retinal degeneration and macular degeneration. I told you about its effects on blood pressure. An interesting one is pulmonary hypertension. Babies in utero perfuse the placenta to get oxygen from the mother's blood in the placenta. Their lungs are poorly developed. If they're premature babies weighing 1 or 2 kilos, their lungs are not developed. They have pulmonary hypertension. They shunt blood right to left through the patent foramen, through the patent ductus arteriosus. They're hypoxic blue babies, and they usually die within a couple weeks. If they inhale nitric oxide, they dilate their pulmonary vessels. There's less shunting right to left. They now become better oxygenated, and they survive. It's been marvelous. Almost all neonatal hospitals, units in hospitals around the world now use inhaled nitric oxide. Some of them, I suggested, should also use a phosphodiesterase inhibitor like Viagra. And one time a Turkish neonatologist, I was giving a lecture in Tunisia, and a Turkish neonatologist came up and said, "Thank you for nitric oxide. It's doing wonderful things for my babies. However, it's very expensive. I can't afford it. They charge too much." I said, "Nitric oxide's cheap. What's expensive is the machine, because you have to control just the right amount to deliver the babies. Too little won't work. Too much is going to be toxic." I said, "Why don't you give the babies a little Viagra? That'll decrease the cost of nitric oxide." So many hospitals are now doing both. I told you about penile erection. This is of interest to you. The blood vessels in the corpus cavernosum have nerves that release nitric oxide as a neurotransmitter. The nitric oxide increases cyclic GMP. It causes relaxation of the blood vessels. The blood vessels fill, and that's the mechanism of penile erection. It plays a role in angiogenesis. It promotes endothelial cell proliferation and inhibits smooth muscle proliferation, so it forms microvessels to feed the tumor. And the tumor is talking to the blood vessels to do angiogenesis, because the nitric oxide in the tumor can't make cyclic G because the guanylate cyclase is defective. So the nitric oxide comes out and enhances angiogenesis to feed the tumor, and that's how tumors survive. If you can interrupt that pathway with things that regulate nitric oxide or cyclic G, you now have new therapies for cancer, and that's something I've been working on for the last 10 or 15 years. Plays a role in atherogenesis. These are inflammatory diseases. Plaque formation is influenced by cyclic G and nitric oxide. Any inflammatory disease has free radicals, reactive species, and that's going to influence nitric oxide production and activity. Next slide. This is my last slide coming up. Plays a role in tissue transplantation. It inhibits platelet aggregation. It influences gastrointestinal motility. It influences ion transport in the renal tubule. It regulates hormone secretion from pituitary, from the beta cells in the pancreas. It regulates insulin secretion. It regulates growth hormone secretion. It regulates ACTH production and secretion from the adrenal. We know it regulates genes. It really is interesting with gene regulation. And we've been beating on that problem now for about 10 or 15 years, trying to find the target that nitric oxide regulates and cyclic GMP regulates in gene regulation. And should we find out that target, hopefully we'll find some new ways to influence transcription in cancer stem cell differentiation and proliferation. And I think stem cells have a great future and may replace some of the tissue transplantation needs that we have. Let me stop at this point. I've probably said too much. I hope I didn't confuse you. I want to thank you again for inviting me. It's been a fun area of research. It's very rewarding to see that it has so much medical application. That's very exciting to me. Thank you very much for inviting me.